Thank you very much, Franco. I just wanted to say that for all the 4 panelists, we'll have questions at the end of the session. And with that, I'd like to invite Professor Xi-Nian Zuo, who's a professor of developmental population neuroscience at State Key Lab of Cognitive Neuroscience and Learning at Beijing Normal University and an IDG McGovern Institute for Brain Research, Development Population Neuroscience Research Center at Beijing Normal University. He's also a fellow of the Organization for Human Brain Mapping. Dr. Zhu has led multiple large-scale data sharing projects, including the Consortium for Reliability and Reproducibility, the 3R-BRAIN and Chinese Colorness project. He's been responsible for building the interdisciplinary brain database for in vivo population imaging at National Basic Science Data Center. I'd like to invite you to the stage.
Well, thanks for the invitation, and also I'm very grateful to be here and sharing our experience of, increasing the diversity of data just mentioned in last talk. And today I'm going to talk about the 3R-BRAIN consortium. Okay, so we all know there are lots of threats to reproducible science, and now it has been well recognized we should do open science and also pre-registration studies and team science. So, but we use these terms for reproducibility, without definition, formal definitions. And then here I introduce a formal definition for these 3 R's. So the reproducibility is reperforming the same analysis with the same code using a different analysis. Replicability, reperforming the experiment and collecting the new data. Changes of the study elements would be experimental data and also analysis, code, estimate, and claim. For reliability, it's a special form of replicability for the question of individual differences with the same analysis and individuals. So the replicability of individual variability across different occasions. Regarding the psychometric theory, both reliability and validity were defined based on the individual differences. So from the screen, you can see the validity is the true disorder or traits related to the questions you're asking. But the reliability is included many contaminants unrelated to the questions you're asking. So in psychometric theory, validity is less than reliability, but we never know the validity. We know how to measure reliability. So this is why we need to harness reliability for neuroscience research, and also this is very critical for our experimental design. So this is there are some facts from theory. The effect size we observed is reliability weighted the true effect size. We can't measure directly. So given a effect size, studies with more reliable measures would need less samples. So given measures with almost perfect reliability, 300 samples can offer a study 80% power to detect small effect size. The given measures with slight reliability means less 20%. That we would need big data, more than 1,000, to detect a small effect size. In terms of these facts, so if we really need to deep phenotyping such vulnerabilities, means we need representative individual differences. And we have now in the field, we have lots of large-scale data sets. Means we can have many subjects, and within individual we can perform really long data acquisition for each individual. But for such data sets, we can use them to build a normative individual differences. But if these measures are not reliable enough, and then the results would be not robust to reproducibility. So there are some, I put the lifespan brain charts here. This is based on the morphological measure. Those measures are really reliable. But for some functional measure, not reliable, it would be very challenging to build such brain charts. So to do the deep phenotyping variability, we really call for psychometric studies. But in this field, now we know this, but we also do have computational resource to calculate this measurement of reliability. But we do not have large-scale data sets for psychometric studies. This 10 years ago, we released the data by Consortium for Reliability and Reproducibility, which has been widely used for the field to do the psychometric studies. So in this talk, I will introduce the 10 years effort. After that, we built the 3R-BRAIN resource to perform a multi-stage deep phenotyping and get richer diversity of psychometric design. This resource includes 3 stages. The first stage is a deep design for retest diversity. We recruited 100 participants. Each participant was scanned 5 times across 3 scanners. In the 2 scanner, 3 Tesla scanner, we did a retest design, separated by 2 weeks. On the right side of the screen is the final outcomes. We finally got 392 scans from 103 participants, including their brain imaging measurements and also psychometric measurements. For the second stage of 3R-BRAIN, we go for a deep design for stimulation diversity. We recruited 50 participants and for 500 scans. Each participant watched 5,000 pictures in 5 to 10 hours resting functional MRI. So they are, on the left side is the details of the design. Oops, I missed something. Okay. So for the last stage of this resource, we performed a deep design for platform diversity, and we recruited 50 participants and for 500 scans. Actually, we do not have such data for the same group of subjects travel to many different scanners for this in the present time for the field. This one we want to share to the field. So the design is only performed at Beijing, so which we call it a Peking Travel Club. So we finally got 9 scanners plus 1 MEG scanner. So we did the resting state fMRI scanning with 2 conditions, including eye closed and eye open. So the order of these 2 states are counterbalanced within individuals. So we used multi-echo plus multi-band sequences to acquire these 4 15 minutes resting state fMRI scans. Below are the 5 sites. The first is the Chinese Academy Institute of Biophysics. You can see they have Prisma 3 Tesla and Altera 7 Tesla. Okay. The second is Peking Union Medical College. They have United Imaging MRI Jupiter, which is a 5 Tesla scanner, plus 793 Tesla scanner. The third one is the National Clinical Research Center for Neurological Diseases. They have Terra Plus 7 Tesla scanner plus United Imaging MRI NX 3 Tesla scanner. The fourth is Beijing Tiantan Hospital, affiliated Chinese Medical University. They have Prisma and 880 3 Tesla scanners. The last one is Beijing United Imaging Research Institute of Intelligent Imaging. They have 890 scanners, 3 Tesla, which is. So finally, we got these data sets almost done now, which you can see these the 9 scanners and also the imaging sequence settings. So finally, I want to show you the individual variability of this is the cortical thickness from one volunteer traveled across the 9 scanners. On the right side, hopefully you can see the movie. It seems not moving, okay, but we still can see there are pretty much variability across the different scanners. And also for the population, it is much better. Okay, it is moving now. So, but around the boundaries among different areas, we still can see the these variabilities across scanners. So for functional MRI, we can see, just to give you one example, these are used for multi-echo. We can see this increase of signal-to-noise ratio. So this data will be released at the Science Data Bank by 2 weeks. So thanks to the technical support team and also my team, I show you the 2 working house members. Thank you very much.